So, hi everyone. Can everybody see the slides? Yes, I think you can.
Yes, we can.
So, thank you for the opportunity to speak, Martin, and congratulations already ahead. Wait, I think I'm on the wrong slide. Sorry. So I won't be able to, in the few minutes that I had, highlight all the exposome related studies. So I will highlight the ones that I've been involved in and the most, and but from a distance, because Dr. Sylvia Sanchez and Dr. Sarah Daniels were on the field in a study that leverages the Lollipop study. Together with Stephen Rappaport, Martin reached out to Professors John Chambers and Jasper Kuhner at Ealing Hospital at Imperial College, and they embarked on an exposome study to investigate the environmental and genetic factors and mechanisms underlying heart disease, stroke, diabetes, and obesity, and anything other major medical problem, basically. And this was done by leveraging those ongoing studies at Ealing, which are called the Lollipop, which basically focused on a diverse population of Indian Asian ancestry that migrated to the UK and had higher risk of, for example, heart disease. And so the idea was to tackle an exposome approach to address these questions. And importantly, when the field team came back, we almost didn't dare touch these precious samples because it had not only taken us so much time to prepare and optimize how to collect these, but when the field team got there, nothing was in place. This hospital had never collected biological specimens in any whatsoever manner, so they had to improvise, and they still succeeded to get high quality samples that were not thawed over liquid nitrogen or dry ice when they were shipped to us over the Atlantic. So we barely dare touch them. So we started a stability volunteer study because we were also not sure, like, what if we find something that is exposome? How do we know that this is really something that is stable over time and that definitely contributed? What if it's a one-time thing because somebody ate some fish yesterday and that's what we're picking up? So we needed some kind of reference material. So we started at Berkeley our own volunteer study to really, like, with 6 time points within a year, the same volunteers came back and donated all kinds of biospecimens. And we were able to start looking, for example, at microRNA and the stability in those samples over time, but also to be able to account for technical and experimental variability, like extraction, like how do these components contribute and make noise in all this data, because the exposome data is already noisy. So that was one of the studies I was very heavily involved in at my time at Berkeley, and I think we still use these samples. There's still much more publications that could come out of these, so they're still very precious, both the Lollipop samples and these volunteer study samples. So there was a massive team, both on the field and in the field, but also here back in, or not here, but back in Berkeley in the lab, that was involved in one way or another with the exposome studies, or with the volunteer study, and with the Superfund study, because we did it all. And if Martin had an out of the box, let's not say crazy idea, we would jump on it and we'd do our best to address and try to address the questions and come up with solutions. And so here is a reflection of all the people, and this is not even all of them, I couldn't get pictures of everybody, but there was a massive team involved and passionate to really work on this. So one of the students, because we were allowed to grill Martin in our time here, articulated very well our transformative time with Martin in the lab. So from this transformative experience blossomed year-long relationships that can be best summarized by Charles Dickens. It was the best and it was the worst of times. The worst of times, because how could one expect that the next research idea Martin would have would entail an overnight confinement to a cold room. So as you can see here down here, is that wearing winter jackets and not even be warm enough to do an experiment in the cold room. Or, but one could always count on Martin to stop by our posters at every conference, even if it was just to highlight the errors that we had on the poster. And then one student who especially had a long tenure with Martin sent a nice comment. So I wanted to highlight that was Dr. Sylvia Sanchez. She said, exactly 10 years ago, you sent me to London for my first Exposome adventure. You told me in January that I would be back in 4 weeks. And I didn't return home until June that year. So they stayed for 6 months. This was their first time abroad and the first time in the winter and all that stuff. So it was a very unique experience. But that Exposome project motivated her to pursue higher education and has opened other avenues for her to continue actually until today, Exposome related research. And she said, although I was ill prepared for this experience, I emerged as a highly organized, hardworking, and better manager, student, and researcher than I was before my time with Martin. So, and personally, I got excited about Exposome research through Martin and Steve Rappaport's vision. And I'm currently directing the Exposome Collaborative here at Johns Hopkins University. So the Exposome will go on. And the best of times were clearly highlighted by the fact that we could always count on you, Martin, and Looping to start the dance party at every conference that we went to. You would help students crash receptions. You would start a snowball fight. So I heard somebody said there was a shoe fight. Well, in this snowball fight, Martin was actually a participant, and that was actually at Ealing in the UK. And you would always let us go all out for lab dinners with your credit card. There were no restrictions. Or you would make things more entertaining by making bets on how much money it would take for any of us to jump into the pool. Martin, I think we would like to thank, from all of us, we would like to thank you for the wonderful times we shared throughout these years, for the lifelong friendships that we were able to make in your lab, for the dark humor that we inherited from a Brit who doesn't even like the UK, and for igniting our passion for good science and for the many opportunities that you have made possible for all of us. Congratulations and keep partying on.
Thank you, Fenner. Unless there are any other comments, we'll turn to Professor Emeritus Steve Rappaport to do a summary and wrap up. Steve's a long-term colleague, collaborator, and friend of Martin's. Let's see. Where do I find this thing? Oh, okay. That's that one, maybe? No, it just says Rappaport. So it's in session 2. I don't know where